Okay, today we're going to look at paper 6, which is the alternative to the practical for biology. And that's for IGCSE, which is from Cambridge International Examinations, CIE. And that's from May-June 2018, and the code is 0610/61. All right, let's get started.
Oh yeah, let's light this candle.
Question 1. Young mammals feed on milk containing protein. Some mammals produce an enzyme called renin. Renin changes the protein in milk so that it can be digested by another enzyme. The action on renin causes small lumps or clots to form in the milk. An investigation was carried out to find the effect of pH on the activity of the enzyme renin. Step 1. 3 test tubes were labeled P, Q, and R, and a syringe was used to add 5 centimeters cubed of milk to each of these test tubes. Step 3. A dropping pipette was used to add 2 drops of acid to test tube P. A dropping pipette was used to add 2 drops of distilled water to test tube Q, and a dropping pipette was used to add 2 drops of alkali to test tube R. Another 3 test tubes were labeled P1, Q1, and R1. A clean syringe was used to add 1 centimeter cubed of 0.1% renin solution to each of test tubes P1, Q1, and R1. Step 8. All 6 test tubes were placed into a water bath at 40 degrees Celsius and left for 3 minutes. Step 9. The contents of test tube P1 were added to test tube P. The contents of test tube Q1 were added to test tube Q. And the contents of test tube R1 were added to test tube R. Step 10. Test tubes P, Q, and R were kept in the water bath and a stop clock was started. Step 11. After 1 minute, test tube P was removed from the water bath. It was tipped and rotated as shown in figure 1.1. Okay, this is this diagram right here. The appearance of the milk was observed and the stage of clotting was decided by comparing it to the diagrams in figure 1.1. So stage A, there was no clotting, and basically if they twisted the test tube, the milk would drain back smoothly from the sides of the test tube. So it was basically milk. Stage B, there was a little bit of clotting, some clotting. The small clots stick to the side of the test tube when they turn the test tube. And stage C, it was completely clotted, all clotted. Most of the milk is solid and does not pour when the test tube is tipped. Okay. Then step 12, test tube P was returned to the water bath. 13, steps 11 and 12 were repeated for test tubes Q and R. So just as a refresher, 11 and 12, after 1 minute, test tube P was for Q and R. Test tube Q and R were removed from the water bath. It was tipped and rotated as shown in figure 1.1, and the appearance of milk was observed. And the stage of clotting was decided, and then it was returned to the water bath. So basically, so steps 11, 12, and 13 were repeated every minute for 5 minutes. So basically, after the 3 minutes that all 6 test tubes were put into the water bath and reached the same temperature, the renin was added to each one of the different situations, either acid, alkali, or neutral, of the test tubes. Okay, and then basically every minute they were taken out after that to test to see whether or not there was clotting or not. And the reason why test tube P was returned to the water bath but 11 and 12 were repeated for test tubes Q and R is due to the next step, because the results are shown in figure 1.2. Test tube P had some clotting at 1 minute and was all clotted at 2 minutes, so basically that means that they didn't need to keep testing it. So that's why it was only Q and R that was tested. Test tube Q had no clotting at 1, 2, or 3 minutes, but some clotting at 4 and 5 minutes. Test tube R had no clotting throughout the investigation and remained unchanged after 5 minutes. And then, finally, prepare a table in which to record these results. Use the information in figure 1.2 to complete this table. Okay, so for these exams, they always start off with a question that's some sort of experiment. And it can sometimes be a bit confusing to figure out what they're asking at first. So they're generally not complicated questions, but there's a lot of steps. And what you have to do is go and read through the question probably 3 or 4 times to make sure you fully understand what is going on. Because generally, after the first time you read it, you don't have a clear enough picture as to what's happening. So you have to read through it a few times, highlight the important parts if you need to, but make sure you know what is happening. Okay, so now we need to prepare a table in which to record the results. And we need to include the information in this table here. Okay, on this box here. Okay, that's what needs to be included. All right, so how do we do that? Okay, so what sort of information do you need to include? You need to include that there's test tubes. You need to include that there's test tubes, P, Q, and R. This is just my rough working. This is not the answer to the question. Okay, you need to record what happens at times 1, 2, 3, 4, and 5 minutes. And you need to record the stage of clotting, whether it's A, B, or C. Okay, that's a lot of information, and you could do it in some massive table where you have many, many columns, but that's not good. You want to make it as simple as you can. Okay, so how do we get all that information into one neat and easy to read table? Okay, this is the way that I would do it. Okay, so the first column you always put the independent variable, the thing you're changing. And so that in this case is the test tube. Okay, and the second column is the time. For this one, it's time in minutes. It's very important you put the units. Okay, you will lose marks if you don't put the units. Okay, in the third column, you can put the stage of clotting. And is that A, B, or C? Because that was specified in the instructions. Okay, in the test tube, it doesn't have units. You don't have to say whether it's acid, alkali, or neutral, because in the instructions it specified whether it was what test tube P, Q, and R were. Okay, so in biology, it's very important when you draw a table that you also use a ruler for your lines, and you draw all the cells, including the outside. Okay, so I'm going to start filling in some of this information. We'll fill in the results in a moment. Okay, so here's the table I would do. So in the test tube, we have test tube P, Q, and R. Okay, so this is time at 1 minute, 2 minutes, 3 minutes, 4 minutes, 5 minutes, time at 1, 2, 3, 4, 5. There's other ways you could do the table, but this is a good way of doing it. And now we have to put in the stage of clotting. Okay, so test tube P had some clotting at 1 minute and was all clotted at 2 minutes. So some clotting was stage B, and all clotted was stage C, and so it stayed at C for each minute after that. Test tube Q had no clotting at 1, 2, or 3 minutes, but some clotting at 4 and 5 minutes. And test tube R had no clotting throughout the investigation and remained unchanged after 5 minutes. So no clotting at 1, 2, 3 minutes, but some 4 and 5. So that was stage A, then B, B, and test tube R was A all the way through. There was no clotting at all. Okay, part B. Now we need to state a conclusion for these results. All right, to look at this, test tube P was acidic, test tube Q was neutral, and test tube R was alkali. Test tube P, the renin worked very well because it's meant to clot the milk. Test tube Q, it worked slowly, and test tube R, it didn't do anything. Okay, that's basically what we need to write for our conclusion. So, renin's optimum pH is acidic. And when you're talking about enzymes, they like to see optimum pH or optimum temperature. Those are nice keywords that they like to see. Okay, so when it's neutral, it acts more slowly and does not form clots at all when it's alkaline. Okay, so renin's optimum pH is acidic. Renin acts more slowly in neutral conditions and does not form clots in alkali conditions. All right, good. Part C1. Suggest why, in step 8, all of the test tubes were placed into a water bath for 3 minutes before the contents were added together in step 9. Before mixing the contents together in step 9. Okay, the reason for that is because it gives time for all the solutions to equilibrate, for them to all reach the same temperature. Okay, so this allowed them all to reach the same temperature. C2, state 2 variables that were kept constant in this investigation. There are obviously many variables that are kept constant, but they only want 2. If you put more down, then if any of them are not perfect, if they're not exactly what they're looking for, you will lose a mark. They cancel each other out. So only put down the number that it asks you to put down. Okay, so the 2 that I put down were volume of milk and the concentration of enzyme. You cannot just put down the word volume though, or concentration, because you need to specify what volume was kept constant or what concentration was kept constant. Okay, other things you could have put down, you could have said the volume of the acid, you could have said the volume of the enzyme, you could say the temperature, because it was a water bath, you could have said the total time that it took, or the equilibration time. You can't just put the word time down, okay? So you have to be specific with these variables. So the total time or the time for equilibration, but not just time. Okay. D, identify 4 sources of error in this investigation. No investigation's perfect. Some are better than others, but there's quite a few sources of error in this one. Things you cannot put down. You will not get any marks if you say something like human error. That is a big no-no. That's not a source of error. That is just, yes, it happens, but it's not a specific thing that you can change next time. Okay, and you can't put down things like read the thermometer wrong. Okay, that's very much like the first one, human error. Those are things you just don't include. You cannot include. Okay, because you can't change the next time. There's always a chance you could read it wrong, but that's why you do 3 tries. You repeat investigations to make sure you get rid of the human error. Well, you don't get rid of it, but you minimize it. Okay, one of the big things that is a source of error in investigations such as this is that you're just looking at something and saying, well, that looks about that. It happens in pH as well. When you're looking at a pH strip, you say, well, that's kind of yellow, I think. So how you word it, if you're saying that it's basically you're guessing if it's close to a particular point, you say it's subjective. Okay, it's a subjective observation. Okay. Okay, another big source of error in this one is they only did the experiment once. You all know that the way you always get a free mark when you're designing an experiment is you always write down, repeat the experiment at least twice. So you have to do basically three trials just to make sure that if there is a human error in there, you know it's there. Okay, so no repeats is one of them. Another thing was that they used dropping pipettes, and dropping pipettes are pretty good. You can be quite precise with them, but it depends on how precise you're trying to be, because dropping pipettes, sometimes the drops are bigger than other times. Okay, so that can make a big difference. You could say the drops of the acid or the drops of the water or the drops of the alkali could be different volumes. Okay, so the drops of the acid, the alkali, and the water could be different volumes, and that could make a big difference if you're trying to use the same concentrations, but you have more acid than you do alkali. It's not a fair test. You could also say that when they tipped the contents of P1, Q1, and R1 into P, Q, and R, some of the contents would remain in the test tubes. Okay? Because as you know, if you pour one thing into another, there's always a residue on the inside of the test tube. You don't know if it's the same amount. Okay, so some contents remain in test tubes when adding to other test tubes. Okay, other things you could have put down. The 3 test tubes were not observed at exactly the same time. You could say that the tip and rotate method that's described wasn't exactly standardized. And a big one that they might like to see is that there was no control. So remember, the control experiment is when you don't put any of the enzyme in, and it would show what happens to milk without the renin. So if you put the milk in the water bath and after that time it started clotting, well, then your whole experiment's out the window. Okay, so the control experiment would show that milk does not clot without renin. Okay, so any of those would work as sources of investigation. Okay, E. Identify one hazard associated with this procedure that would require the use of eye protection. Well, for eye protection, they're talking about splashing generally. And the things that could splash are acid or alkali or the enzyme. None of those you want in your eyes. So just choose one of them. There's only one mark, and it only wants one hazard. So just choose one. So you could say the use of the acid. Good. And that's all it wants. Part F. Clotting separates milk into a solid part and a liquid part. Describe how you could find out if there was any protein remaining in the liquid part. Okay, the test for protein is the Burette test. So you add Burette to the liquid part, and if it turns purple or lilac or mauve, then it is positive and there is protein. Okay, so you add Burette to the liquid part. If it turns lilac or purple or mauve, then it is positive. And positive means that there's protein in it. So the 2 marks for that, one is the word Burette, and the other is that what happens if it's positive. Okay, that it changes color to a lilac, purple, or mauve color. Okay, G. State the name of the test that would be used to test the milk for the presence of fat. Okay, and that is called the ethanol emulsion test. So when you add ethanol, if there's fats in it, then it forms an emulsion. So it becomes cloudy. Okay? Good. H. After renin has changed the protein in milk into a white solid, protease enzymes can be used to digest the protein. The digested protein forms a colorless liquid. A hypothesis stated that the optimum temperature for protease enzymes to digest changed milk protein is 37 degrees Celsius. Body temperature. That's an interesting hypothesis. State a method that can be used to test this hypothesis. All right. So the first thing you'd want to do is you'd want to test it at different temperatures. Well, one of the temperatures you definitely want to test would be 37 degrees Celsius. But you'd also want to test a temperature a little bit above and a little bit below. Okay? You probably want them fairly close in value, but you don't want it 36, 37, and 38 degrees because that would be really difficult to maintain. Okay, so first of all, you'd want to use 3 temperatures at least. You could say use 5 temperatures as well. That's also a good number. I'm going to say 34 degrees Celsius, 37 degrees Celsius, and 40 degrees Celsius because you definitely need one at 37. And you can't have them too close because you just can't really keep those monitored. Okay, so next thing is, in order to keep them at that temperature, one of the keywords that they like to see is thermostatically controlled water bath. That's one that that the examiners like to see. And that's basically an electric water bath, which is a lot easier than trying to maintain a Bunsen burner a beaker on a Bunsen burner with a thermometer in. It's much more difficult to keep that at a constant 34 degrees Celsius. Okay. So another thing you'd want to do after that, you'd want to use the white solid or the clot from the same sample of milk. You don't want to take it from different samples of milk. That would be silly because you want to test the same thing. Okay, you'd also want to use the same pH for all the different solutions. After that, you'd want to equilibrate temperatures of enzymes and milk solution before mixing, just like they did in the experiment described. Okay, then you time how long the samples take to become colorless. The shortest time has the optimum activity. And another 2 things you always want to put down when you're describing a method for an experiment, you always want to say, repeat at least twice. So when you say repeat at least twice, it means do the experiment at least 3 times. You do it once and then you repeat twice. So that's 3 times in total. And another thing you always want to put down is a safety measure. So generally, when you're talking about things that could splash, you're talking about eye protection or goggles. Wear goggles. Okay, so the complete method in bullet points, because that makes it a lot clearer for the examiner and for you to make sure you have enough marks. Use 3 temperatures, 34 degrees Celsius, 37 degrees Celsius, and 40 degrees Celsius. Use a thermostatically controlled water bath. That's one of the key words I like to hear. Use the white solid from the same sample of milk, or the clot from the same sample of milk. Use the same pH for all the different solutions. So basically you're using the same. You're trying to set up the things that are the same, the controls. Equilibrate temperatures of enzymes and milk solutions before mixing, another control. Time how long samples take to become colorless. The shortest time has the optimum activity. You're saying what you're measuring there. And then you repeat at least twice and you wear eye protection. Okay, so that is 6 marks in total. And here there's 1, 2, 3, 4, 5, 6, 7, 8. There's 8 bullet points. You probably don't need all of that, but all of those are good options. You might want to include 8 in there. We never miss out a method without saying repeat at least twice and a safety measure. You might want to include all 8 of them, especially repeating at least twice and wearing eye protection, because sometimes, as in this particular case, the controls, you'll only be allowed 2 or 3 marks. For this one, it only allows 2 marks from either the same pH of solution, same concentration or volume of enzyme, or making sure the temperatures are at equilibrium. Okay, so that you might put down 3 things, but they're only 2 marks. Okay, so for a method, you might want to just include a few more marks. As many as you can think of, but not going crazy. You only have a certain amount of time in your exam. Okay, question 2. A student wanted to investigate a garden ecosystem. She counted the number of insects caught in spider webs in one small section of the garden. She found 6 spider webs in the small section of garden sampled. Diagrams of the spider webs are shown in figure 2.1. Each black dot represents one insect caught in a spider web. Okay, A1, use figure 2.1 to complete table 2.1. Okay, so basically they want you to count how many spiders were caught in each of the webs. Okay, so let's do that. Okay, web A had 1, 2, 3, 4, 5, 6. Web B had 1, 2, 3. C had 4. D had 1, 2, 3, 4, 5, 6. E had 1, 2, 3, 4, 5, 6, 7, 8. That's a busy spider there. F had 1, 2, 3, 4, 5, 6, 7, 8, 9. Also a busy spider. Okay, now we just fill out the table. Okay, so A was 6, B was 3, 4 was C, D was 6, E was 8, F was 9, and the total, if you add them all up together, it is 36. All right, good. That should be nice and simple. Those sorts of marks are the ones you want on exams. Nice and easy. 2. Calculate the average number of insects per web in the small section of garden using the information in figure 2.1 and table 2.1. Okay, so for the average, we need the total number of insects divided by the number of webs, which equals 36 insects divided by 6 webs. So that is 6 insects per web. Okay, you probably don't need the units in there. However, it's always good practice to put them in anyway. Okay. 3. The student counted the total number of spider webs in the whole garden and found that there were a total of 102 spider webs. That's a lot of spider webs. Use this information and your answer to part 2A2 to estimate the total number of insects caught in webs in the whole garden. So we have 102 webs times 6 insects per web. That equals 612 insects. That's a lot of insects. Again, you probably don't need to write the units in there of insects, but why not? Okay, 4. Suggest one reason why the estimated total number of insects caught in webs in the whole garden may not be accurate. Okay, now this is important. Only put down one, no matter how many you can find. Okay, so one thing you could do is that you could say that there's a huge variation in insect populations in different parts of the garden. So imagine there's probably not as many insects in the part close to the house where you do a lot of walking, and there might be a lot more spider webs down at the far end of the garden where you're not going to be, we're not going to be going very often. Okay, so there's a large variation in insect populations in different parts of the garden. That's highly likely. Okay, other things you could put down is that there might be some of the spider webs might be hard to see. You might be able to say that some insects were eaten by the spiders or they fell off the web, so you didn't count all of the spiders, all the insects that were there. You could say that some of the insects were too small to be seen, or maybe they were difficult to count, maybe they were clumped together. You might say that some caught organisms may not actually be insects. Maybe they were leaves that got twisted in the spider webs. There's lots of things you could put down there. As long as you only put one, then that should be good. Okay. B. Figure 2.2 is a photograph of a spider. A spider's body has two main parts. The legs are all attached to the cephalothorax, which is the upper part of the body, basically around where the head is, and starts at label X. On figure 2.2, the lower part of the body is called the abdomen and is nearest label Y. So the cephalo meaning head, thorax being the middle bit. So this is one section, and this is the other section. This is the cephalothorax, and this is the abdomen. Make a large drawing of the spider in figure 2.2 to show its outline, including its legs. Label the abdomen. Now, there's always a drawing question on a biology practical or alternative practical exam. Now, and it should be easy. Quite often they're simple. This one's not actually as simple, but it's okay. But what's really important is there's a few key things. One is you are not an artist when you come to a biology class. In biology, you want to draw simple lines. Okay, if you draw a circle, you have to draw one clear circle where the start and end touch. This is not a circle. If you draw a cell like that, you will lose marks. Okay? Another thing, you do not feather. You're not an artist. You're not shading. You're not doing anything like that. You're doing one smooth, solid line where things start and end touching. Okay? Other than that, if you are a terrible drawer, like I am, as you'll see in a minute, it's okay as long as things are the same proportion. Okay, those are some very key important things. Do not feather. This is feathering. Do not shade using your pencil, which I can't do with this tablet that I'm using. You can't leave open gaps if they're not really there. Okay, so you just draw the outline. You want to draw the abdomen. Okay, you draw cephalothorax as best you can. Okay, I think that's about it. And as you can see, I'm not an artist. I haven't drawn any details on the body. There's a bit of a head because it says to draw the outline. I haven't drawn any dots because there's some patterns on the body. You don't do that. You're only drawing the outline. Another few things you want to do, you want to make sure that your drawing doesn't cover up the words above or below. You need to take up the space, but you need to take up the majority of the space. Okay? When you draw the legs, as you can see, well, as you know, spiders are arthropods. Arthro meaning jointed, pod meaning foot. So they have jointed legs. You need to draw in that they have jointed legs. Okay? You need to draw in that they have their spinnerets and these little bits here, the chelicerae. And you also need to read the question again to make sure you've included any labels. It says, make a large drawing of the spider in figure 2.2 to show its outlining, including its legs. And you would be surprised at the number of students who don't read the next little bit. Label the abdomen. Okay? For some reason, the way that's placed, people often miss that little statement there. So the abdomen, using a ruler, there we go. Now, when you do label lines in biology, it's just one straight line. You have to make sure if you draw two lines that they don't cross. That's not a good thing. And there's no arrowheads. An arrowhead is not a label line in biology. Okay, so you just draw it to to the structure and have it touch it. Okay? All right, good. And okay, so there's 5 marks in this. You get 1 mark for making sure that there's a single clear continuous line without any shading. So if you do any feathering or any shading, you lose that mark. And you have to try very hard to make sure all of these little bits touch. Okay, I tried really hard. I might have missed something, but I tried hard to make sure that they all touched. Okay, that you want it, 1 is for size. You want it to occupy at least half of the space provided. It must be at least 75 millimeters in length. I don't know how long this is. Okay, the detail, you need to make sure 2 body parts and 2 chelicerae or 2 spinnerets are included. So basically you draw what you see. They're part of the outline. And you need to make sure you have 8 jointed legs attached to the body in the correct position and the correct orientation. So you don't just draw your little outline of your body and say, well, there's 8 legs. You know, that's sort of right. That's a terrible drawing anyway. But you have to make sure that they're going in the same direction as it started with. Okay, and you need to make sure you have your label line, and it's an actual label line. It's going to the abdomen, which it even told you where it was. Okay, that's good. 2. Measure the length of the spider between points X and Y on figure 2.2. Include the units. Okay. So I have mine printed out on a piece of paper. It's a different size to the paper that they use for the exams. And mine came out as, when I measured the line, length X to Y, I measured 36.0 millimeters. Okay, so mine was 36.0 millimeters. But the mark scheme says it's 38.5 millimeters to 41 millimeters. So there is still a little bit of variation they accept, even though there is a specific size of it. So, but you do have to be careful. Okay, but if you're doing it at home, don't panic because you probably did it on a different size piece of paper. Okay, so you have to draw a line in the same position on your drawing and measure the length in your drawing. Okay, so I'll draw the line on the same position. So if we look at the original, it goes from the top of the cephalothorax to the bottom of the abdomen, not including the spinnerets or the chelicerae. Okay, so I've just drawn a line from the top of the cephalothorax to the bottom of the abdomen. Okay, now it wants you to measure it. Unfortunately, I can't measure it off of the screen, so I'm going to say it wanted a minimum size of 75 millimeters, so I'm going to say that when I used my ruler, which I haven't, but if I were to use it, I'm going to say that this was 75 millimeters. Okay, but it's not. All right. Okay, so measure the length of line XY on the spider in your drawing. Clearly it was 75 millimeters. As long as you can measure it during the exam. Now we have to calculate the magnification of your drawing using the measurements and the following equation. So magnification is basically the length of the line on your drawing divided by the actual length of the line. Okay, so magnification equals 75.0 millimeters divided by 36.0 millimeters, and that equals, if you notice that, the units cancel out, 2.08. Okay, so we don't normally write 2.08 magnification. Normally it's by a much more whole number, so we could say it's 2. The magnification is 2. That's it. C. Table 2.2 contains some other data collected by the student from the garden ecosystem. So, when they were out there, they looked at different things. They saw different types of organisms, trees, bushes, other plants, herbivores, carnivores, and then they counted how many they saw in the garden ecosystem. So with that information, you need to plot a bar chart of the data in table 2.2. Now remember a few things about bar charts. First of all, obviously, you need to label the axes, just like any other graph. Now bar charts, the bars are separated. And just like any other graph, you should do it in pencil. And you need to use a ruler. Okay? These are very important things that you need to do for all of these. And you need to take up most of the space. Okay, you need to use regular divisions between the numbers, just like for any other ones, but you don't need to start at 0. You can always use a little asymptote. So these are some of the things you need to do for doing bar graphs in particular, but any graph really. Okay, so first of all, we have to start with our axes. Okay, so on the x-axis down here, we have the type of organism. Okay, so on this x-axis, we have the type of organism, and that's trees, bushes, other plants, herbivores, and carnivores. And you want to take as much space as you can. Okay, but remember, this is a bar chart. It's not a histogram, so we need to make sure that there's a gap in between each. So I'm going to say trees take up this space, then bushes will take up this space, other plants will take up this space, herbivores, and carnivores. Okay, that's what I'm going to use for this. All right. They generally give you some sort of logical number of spacings. Okay, so for the y-axis, we want the number found in the garden ecosystem. Okay, so the number found in the garden ecosystem, if you look at the table, it goes from between 2 and 118. So it does look like we can start at 0, and the numbers work nicely that we can go 10, 20, 30, 40, 50, 60, 70, 80, 90, 100, 110, and 120. And if you look at these, each small square would be 2, 4, 6, 8, 10. That'll be 2. All right, good. Now we can plot our graph. Okay, so here's the graph. Okay, we have our axes where they're labeled, and it's nice and even. This is a bar graph, so there's spaces in between each of the bars. Okay, that's very important. You can't have them touching because they're not related. Okay, I have colored in the individual bars, but I would highly recommend on the exam you do not. Okay, in the exam you should do your drawing, all your drawings and your graphs in pencil, because if you make a mistake, then you can erase it. If you color it in, A, you take more time, and B, sometimes it might be difficult to erase it fully. And if they can still see some of the mistake, they may mark that as wrong. Okay, so you need to make sure you don't waste time, and you draw your graphs very carefully, so that if you make a mistake, you can erase it without having the lines showing underneath. And the same goes with your drawings. Okay. They can be very picky with these things. Okay, I think that's about it for the graph. Good. And finally, 2. Herbivores and carnivores are animals. Use the data in Table 2.2 to calculate the ratio of animals to plants. Show your working and give your answer in its simplest form. Okay, when you're talking about ratios, you're talking about fractions, basically. Okay, so the number of animals, you have the number of animals to the number of plants. Okay, so how many animals do we have? We have 118 plus 14 animals to 2 plus 5 plus 37 plants. Okay, so 118 plus 14, that is 132, and 2 plus 5 plus 37 is 44. If we divide 132 by 44, well, we divide them both by 44, which is the smallest number, we get 3 to 1. Okay, so the ratio is 3 to 1 plants to animals, or animals to plants, sorry. Animals to plants. You don't need to write in animals to plants there, but you do double check. In the question it says animals to plants, because if it said plants to animals, it would be 1 to 3, and you'd get the question wrong. Okay, so that's very important. All right, so you've reached the end of this exam. Well done. Hopefully you found this helpful. If you have, please press the like and the subscribe buttons to let us know that you want more. And if you have any questions or comments, please write them in the discussion section below. And have a great day.